Different types of hypersensitivity as type 1, 2, 3, 4, depending upon the response shown by the vertebrate body. Immediate type of hypersensitivity type 1, 2, 3, and delayed type of hypersensitivity come under type 4. So these are the images showing the different types of hypersensitivity. As mentioned in earlier session, when allergen type of antigen bind with the mast cell or basophils, uh, or via the IgE type of the antibodies present on the surface. So this is the allergen specific IgE antibody. Then binding of the allergen to this IgE specific antibody responsible for degranulation of the mast cell or basophil, which is responsible for release of vasoactive substances such as histamines, bradykinin, heparin, slow reacting substances, Hageman's factor, which are responsible for vasodilation, smooth muscle contraction, etc. So the mechanism of type 1 type of hypersensitivity, when there is binding of the allergen with the IgE receptor present on mast cell or basophils, then degranulation or of vasoactive substances from the mast cell or basophil takes place, which are the mediators of these are the vasoactive mediators of anaphylaxis. So typical manifestation include the systemic anaphylaxis, localized anaphylaxis such as food allergies, fever, asthma, etc. Now these are the structure of basophils and mesophils, release of granules containing histamine and other active agents, and how they are released. The antigen or allergen is react with initially B cell, then there is production of the memory cell and plasma cell, synthesis of the complementary antibodies to that allergen, which is allergen specific IgE antibody, then sensitize mast cell with this IgE antibody to which contact of again allergen and degranulation of the vasoactive amines responsible for release of heparin, histamines, serotonin, bradykinin, etc. They accumulate in the smooth muscle cell, blood vessel, mucous gland, and show the response in the type of redness, swelling, will flare reaction on the skin surface, etc. This is the typical structure of IgE antibody. Whatever the vasoactive amines released, they are responsible for showing their effect as increased vascular permeability, smooth muscle contraction, eosinophil chemotaxis, neutrophil chemotaxis, etc. Histamine synthesized from histidine amino acid via decarboxylation, serotonin 5-hydroxytryptophan synthesized from the tryptophan. So such type of substances, these are the mediators of the anaphylaxis or vasoactive amines. So this image shows the vasodilation, formation of the lesions, different types of allergen tried on skin surface, allergen to mite, allergen to grass. So allergies also depend on the factors like geographical distribution, environmental factors. Some person also allergic to sulfa drugs, some antibiotics like penicillin, plant pollens, some proteins like egg albumin, nuts, peanuts. So antihistamine drugs or medicines are used to cure the type 1 type of hypersensitivity. Also epinephrine, cortisone used in the treatment. These are the some images of allergic reaction. Then type 2, it is due to the antibody dependent cell cytotoxicity mediated by antibodies directed towards the antigen present on the cell surface of the extracellular matrix. Non-autoimmune examples of the type 2 hypersensitivity: transfusion reactions of ABO blood group system, hemolytic disease of the newborn, erythroblastosis fetalis. If the father is Rh positive, mother is Rh negative, then in first pregnancy the anti-Rh antibodies are produced from mother. After first pregnancy, anti-Rh antibodies are produced against the Rh antigen of the fetus. If second pregnancy is there and the same embryo having Rh positive antigen, Then the anti-Rh antibodies transferred from placenta to the Rh positive antigen of the embryo, because the initially memory B cell keep the memory of production of these antibodies, and these anti-Rh antibodies cross the placenta and attack on fetal RBCs, causing erythroblastosis fetalis. So to cure this, to prevent this, mother should be treated with the rogum. So this rogum prevents the B cell activation and memory cell formation for production of anti-Rh antibodies. So after delivery, 72 hours, the mother should be injected with such type of immunosuppression drugs, as well as uh, anti-Rh antibodies should be given, at least to prevent the further B cell activation and memory cell formation from mother's origin. So these examples: antibody dependent cell cytotoxicity in type 2 type of hypersensitivity. In type 3, it is due to the immune complex diseases, antigen antibody complex attached with the complement system. They are responsible for showing the effect. The examples of serum sickness, Arthus reaction. In serum sickness, if ready-made passive immunization, active uh, artificial passive immunization for treatment is used as a ready-made anti-tetanus, anti-diphtheria antibodies obtained from serum of horse or sheep. If they are directly administered in the vertebrate body, then the serum sickness arises after 6 days of injection, with symptoms like swelling of lymph node, edema of eyelids, increase in body temperature, etc. In Arthus reaction, if subcutaneous injection of the antigen is administered, the complementary antibodies circulating inside the circulatory system, they react with this antigen. Such type of Arthus reaction possibly observed in case of fungal spore. So this is the antigen antibody mediated reaction in type 3 type of hypersensitivity. So these are the summarized points in allergy or in type 1, 2, 3 hypersensitivity. Type 1, it is due to the degranulation of the vasoactive amines. Type 2, it is antibody dependent cell cytotoxicity. The examples of type 1: asthma, systemic anaphylaxis, fever, food allergy. Examples of type 2 antibody mediated cell cytotoxicity, erythroblastosis fetalis, blood transfusion reaction, etc. In case of type 3, it is due to Arthus reaction, serum sickness, some autoimmune disorders such as rheumatoid arthritis, necrotizing vasculitis. It is due to the immune complex reaction. So this is about the hypersensitivity or allergy.